Once these sites were belatedly discovered, a monarch butterfly biosphere reserve was established, which also received World Heritage status in 2008, SHE 2008. Insect responses to fragmentation of tropical forest and the ecological importance of forest margins also play a role in conservation efforts, e.g. Charnke et al. 2007b. Behavior serves as a central theme in the understanding of the ecology of insect communities in tropical forests, e.g. Bassett et al. 2003, and in the temperate latitudes, see Chapter 12 on community structure and Chapter 13 on multitryphous interactions. There remain considerable opportunities for using the behavioral ecology of insects in the service of humans. Applications rely on increasingly sophisticated technology coupled with the understanding of behavior in life histories of insects. Many natural products, especially in plants, provide repellents and attractants which can be used for pest regulation. Some of these will be discussed in Chapter 4 on plant and herbivore interactions. Summary. Behavior covers all activities in the life of an organism from emerging out of an egg to the last gasp of the dying. All are related to survival, foraging for food, finding or making a place to live, communication and reproduction. Use of experiments to understand behavior is essential. We have covered survival in response to threats from weather, particularly migration, and the challenges of finding food, competition and avoiding being eaten by carnivores. Foraging behavior is complex, requiring a chain of behaviors in a stimulus-response sequence, as we illustrated with a parasitoid searching for hosts for its eggs. In a local patch, time is allocated theoretically according to certain rules such as giving up time, but in reality simple motivations are complicated by many significant factors. Insects need a place to live, which they construct themselves, such as a web or a burrow, or they use places on plants or animals. Behaviors range from females placing eggs exactly where the larva will begin to feed, as in plant tissue, to dropping eggs to the ground from high in vegetation. Communication among individuals and with the environment by insects is frequently accomplished by chemical signals. All organisms release volatile chemicals which act as body odors available to other species for detection. Thus insects of many kinds detect their host plants by chemical signals, and parasitoids may find hosts by chemicals from insect scales, mandibles or frass, or by finding the host plant first and the insect host second. The lexicon for describing the many kinds of chemical communication, or chemical semaphore involving semiochemicals, includes pheromones, allomones, chiromones, cinnamones and apnemones, all of which provide coherent information exchange up and down multitrophic level systems. We treated reproductive behavior under the headings of sexual selection, finding and choosing mates, mating systems, sperm competition, and parental investment. We followed a path from the evolution of differences in form and behavior between males and females of the same species to the tactics involved with finding mates, which may involve hilltopping, lex or territoriality. Courtship enables evaluation of a potential mate, sometimes with tangible nuptial gifts from male to female, while a mating system encompasses a species-specific pattern of male-female association, including mating display, the number of mates, parental care and alternative mating strategies. Sperm competition, when one male displaces sperm in a female from another male, is avoided by many mechanisms such as mating plugs, guarding females, and mating away from meeting places and oviposition substrates. Parental investment is a compromise between increasing the chances of survival of an offspring at the expense of reducing the opportunity to leave more progeny. This often involves parental care of offspring for a period of time, or the careful and time-consuming placement of eggs into high-quality food, or into other well-protected niches. We completed the chapter with a discussion of the application of behavioral ecology to the regulation of insect pests, and its contributions to integrated pest management. This regulation involves such techniques as the use of semiochemicals for disrupting herbivores searching for host plants, increasing cover and food for natural enemies of pests, use of various trapping methods and the sterile male technique. Behavioral ecology also contributes to conservation of insects, as we need to understand migration routes, breeding and overwintering locations, and the best patterns of habitat over the landscape. Behavior of insects serves as a central theme in the understanding of more complex interactions in communities and ecosystems. Questions and discussion topics. 3. Social insects, the evolution and ecological consequences of sociality. Social insects are major components of most ecosystems and are key players in communities. We will see in this chapter that their biomass is impressive, their activities as ecosystem engineers, making nests, trails and moving soil, are massive, and their impacts on other community members are widespread. Social insects stimulate immense fascination among their human observers because of their ubiquity, their diurnal activity and their complex social structure involving many sophisticated behavioral interactions. They also pose the problem of how such societies evolved, under which ecological conditions would selection favor the banding together of related individuals into dense populations distinct from most species whose individuals disperse widely from others. The interplay of life history, evolution, behavior, ecology and phylogeny in the emergence of social insects offers an excellent example of how these biological processes are inevitably meshed together and how we need to address them with an integrated biology approach. Starting in this chapter from basics, we define social insects and their hierarchy of social integration from solitary to highly social groups. Then we briefly describe the ecology and behavior of major taxa in the social insects, followed by an exploration into the likely factors promoting the evolution of complex interactions among closely aggregated individuals. The importance of social insects as participants in community and ecosystem function is then covered in some detail because their impact is multifaceted, complex and strong. Finally, managed lands are colonized by these organized societies, so what we learn about social insects can be applied in agriculture, forestry, horticulture and conservation. 3.1 What are social insects? An insect society is a group of conspecific individuals organized in a cooperative manner. This typically includes helping behaviors, shared resources, and cooperational communication. The shared resources can be shelter, defense, or food. The majority of insect societies are built on the family unit, and how we classify social complexity is largely based on the family relationship. Costa and Fitzgerald, 1996. Indeed, the first real attempt to classify social complexity in insects by Wheeler, 1928, page 12, consisted of 7 categories distinguished by constantly increasing intimacy of the mother with her progeny. The terminology for categorizing insect societies was further developed by Michener, 1969, and Wilson, 1971. Although this social terminology has been criticized for being overly focused on bees, ants, and termites, and for not focusing enough on the trade-off between personal reproduction and cooperation inherent in insect societies, Crespi and Yanega, 1995, Costa and Fitzgerald, 1996, 2005, Wilson's terminology is still the most widely used. Wilson proposed that insect societies increased in complexity from solitary to highly social via the acquisition of 3 key traits: cooperative brood care, reproductive castes, and overlap of generations. Table 3.1. 3.2 Levels of sociality. The other social insects. Solitary insects, of course, show no cooperation among individuals. Subsocial insects, on the other hand, typically form a single family unit where one or more of the parents care for their own offspring, at least for a short time. Parental care is incredibly widespread among insects. It is found in thousands of insect species distributed among at least 15 orders. Ptolemy and Wood, 1986, Costa, 2006. Parental care varies immensely within these species and ranges from passive egg guarding to grooming, feeding, protecting, and nesting behaviors. All of the behaviors, however, can be categorized into those that physically protect the young from danger, those that protect resources vital to offspring, and those that facilitate offspring feeding. Ptolemy and Wood, 1986. Gargaphia solana lacebugs provide an excellent example of defense from danger. These tiny lacebugs feed on plants in the nightshade genus Solanum. Ptolemy and Dino, 1981. Females produce egg masses that are cemented to the underside of leaves, and females remain with their gregarious offspring for all 5 nymphal instars. When predators approach the nymphs, mothers aggressively charge the predators while fanning their wings. Although the lacebugs are not really capable of harming the far larger and more dangerous. Table 3.1. The hierarchy of sociality from solitary to eusocial, with intermediate parasocial conditions. 0 1/4 absence, 1/4 presence. Modified from Wilson, 1971. Components of sociality. Levels of sociality: cooperative brood care. Reproductive castes. Overlap of generations. Solitary, subsocial, and communal. 0 quasi-social, 0 semisocial, 0 eusocial. Predators, lady beetles, lacewings, and the like. The displays are very effective. Nymphal survival was dramatically higher when mothers were allowed to protect their offspring than when mothers were removed. Ptolemy and Dino, 1981, figure 3.1. Not all parental care is provided by the mother. Male giant water bugs, Hemiptera, Heteroptera, Belastomatidae, for example, protect the eggs that they parent by carrying the eggs on their backs. See detailed discussion in Chapter 10. Nigrophorus burying beetles provide a rather extreme example of the efforts of some parents to protect resources for their offspring and facilitate offspring feeding. Ptolemy and Wood, 1986, Costa, 2006. Larvae of Nigrophorus burying beetles use small vertebrate carcasses as their only food resource. Scott, 1998. This resource is essential for reproduction, is unpredictable in space and time, and is valuable to many other animals and microorganisms. Consequently, there is strong selection for adult burying beetles to go to great lengths to secure these resources. Scott, 1998. Once a female beetle or reproductive pair of beetles discovers a fresh carcass, they move it to a suitable spot for burial and dig beneath it. They also remove the hair or feathers on the carcass and start to shape the carcass into a ball. In addition, they clean the carcass and secrete antimicrobial compounds on it, which significantly delays decomposition. When fully prepared, the carcass may be in a shallow burial chamber that is just a depression under leaf litter, or a deep vault that is as much as 60 centimeters underground. When males cooperate with their mates, carrion is buried significantly faster than when females work alone. Wilson and Fudge, 1984. Once all is prepared, the mother Nigrophorus lays her eggs in soil near the carcass, and she feeds the offspring with regurgitated carrion and continues to defend the carrion and developing larvae from competitors, predators, and fungi. Figure 3.2. Frequently her mate will cooperate with these tasks, illustrating biparental care. Communal species are those where members of the same generation live together, but without cooperative brood care. This often occurs in species where there is communal oviposition of eggs, but mothers do not actively care for these offspring. This behavior occurs in a wide range of insect orders. For example, females of many species of Psocoptera. Figure 3.1a. Gargaphia solana female guards her offspring. Although small and relatively harmless, these lacebugs are very effective at repulsing predators. Photograph and copyright by Michael Loeb. See color plate section. Figure 3.2. Parental care by a carrion beetle, Nigrophorus vespilo coleoptera, sylphide, with the adult feeding larva in a nest-like structure. Original illustration by Sarah Landry, based on a photograph by Erna Pukowski. From Wilson, 1971. Book or bark lice will oviposit clutches of eggs in the same location, and, upon hatching, groups of several dozen to several hundred nymphs stay together while grazing on lichens and algae. New, 1985, Costa, 2006, Requena et al. 2007. Most teneral adults disperse within a few days of molting. Requenate al. 2007, and although some females may be found near aggregations of nymphs, they do not protect their offspring and fly away when threatened. New, 1985. Aphids also benefit from living in colonies. Aphids living in colonies are often closely related to parthenogenesis and benefit from group living by two mechanisms: alarm signaling and induction of metabolic sinks in their host plants. When attacked by predators or parasitoids, aphids emit cornical droplets containing the alarm pheromone E-cis-farnesene, Kislow and Edwards 1972. This alarm signal increases aphid survival by causing clone mates to stop feeding and walk away or drop from their feeding sites to escape predation, Nault et al. 1973. Peel and Ho 1970 found that aphid feeding creates a sink in their host plants whereby photoassimilates air concentrated at the feeding site, and this sink effect increases with colony size. Likewise, Lepidopteran species in over 20 different family shave social larvae and can be classified as communal, Costa and Pierce 1997. In many of the species, gregarious cohorts of larvae cooperate in defense, foraging and nest building, but adult butterflies and moths do not care for the caterpillars, Costa 1997. Many tent caterpillars, family Lasiocampidae for example, engage in synchronous rearing and flicking of the anterior half of their bodies when threatened by parasitoids or predators, and use trail marking to facilitate group foraging, Fitzgerald 1976, Fitzgerald and Peterson 1988. Very similar behaviors are found in many species of chrysomelid leaf beetles, Jolivet et al. 1994, and sawflies, Costa 2006. Gregarious sawfly nymphs, for example, benefit from increased efficacy of chemical defense, Cadella Andrafa 1996, and enhanced thermoregulation that results in faster growth rates, Fletcher 2009, as group size increases. Communal living by species of Lepidoptera and Coleoptera often enables them to overcome defenses of their host plants. Caterpillars of the nymphalid butterfly Closyngenize, for example, gain weight twice as fast when they feed in aggregations on their host plants, Odontoma calistachium, than when they feed alone, Dino and Benry 1997. Quasi-social species are socially more complex than communal species because members of the same generation live together and cooperate in brood care. In many allodipine bees in the genus Exonera, for example, multiple sisters will become reproductively active and produce broods in the same nest, Schwartz 1986, Cronin and Schwartz 1999. In several species of Nigrophorus burying beetles, multiple, unrelated females will form cooperative breeding associations and jointly prepare large carcasses and share in the feeding of offspring, Trumbo and Wilson 1993. It is thought that the superabundance of food represented by a large carcass promotes cooperation as a mechanism of reducing the probability of nest failure. In semisocial species, there is cooperative brood care of females of the same generation as well as reproductive caste differentiation. This occurs in some wasps and bees. For example, most Pholistes wasp colonies are semisocial at the beginning of the colony cycle. This occurs when a colony is founded in the spring by a group of females that collectively construct and provision a nest, West 1967. Likewise, Lasioglossum mollicharum sweat bee colonies will persist in a semisocial state when the colony outlives the founding queen. When the queen perishes, workers of the same generation become reproductively active, produce offspring and share brood care within the colony, Richards 2000. 3.3 Eusociality, the superorganisms. Eusocial species represent the ultimate social condition. The majority of the eusocial insects are ants, wasps, bees and termites. These species engage in cooperative brood care, and have reproductive castes and overlapping generations. The key feature of eusociality is the castes. Reproductives are specialized for producing offspring and altruistic workers are specialized for tasks associated with brood care, foraging, nest construction and maintenance, and defense. At maturity, colonies can range in size from a dozen or so individuals up to tens or even hundreds of millions or more, Ho Diaresis LL Dobler and Wilson 2009. Typical eusocial colonies will contain one or at most a handful of reproductives and a much larger number of workers. In ants, bees and wasps, the reproductives are female queens, while daughters are the workers. In termites, however, a king typically lives with the queen and termite workers are often male and female offspring. Eusocial insects typically use pheromones to evoke particular responses in colony mates such as alarm, attraction, assembly and recruitment, although touch and substrate-borne vibrations may also be involved. Most social insects distinguish nestmates from non-nestmates by evaluating cuticular hydrocarbons. Wilson, 1971, has proposed that eusocial insect colonies are superorganisms, due to their integrated communication system, caste-based division of labor and ability to maintain homeostasis via self-regulated, internal feedback loops. The concept of the superorganism is explained by Wilson, 1971, pages 469 to 470, as any society, such as the colony of a eusocial insect species, possessing features of organization analogous to the physiological properties of a single organism. The insect colony, for example, is divided into reproductive castes, analogous to gonads, and worker castes, analogous to somatic tissue. It may exchange nutrients by trophallaxis, analogous to the circulatory system, and so forth. 3.3.1 Bees. Apis mellifera, the European or Western honeybee, is probably the best studied eusocial insect. Honeybee colonies comprise a single egg-laying queen, up to 80,000 workers, and up to 2,000 male drones, Winston 1987. The drone's only job is to fertilize new queens. The nest is an array of double-sided wax combs divided into thousands of hexagonal cells. The hexagonal shape holds the most honey for the least wax and the hexagonal array provides strength. During comb construction and repair, wax is produced by glands on the underside of workers' abdomens and it is gathered and shaped with the front legs and mandibles. Worker bees are sterile females and are kept sterile by the queen who produces a pheromone called queen substance from mandibular glands, Butler 1954. This pheromone is transferred from bee to bee within the hive and restricts ovarial development within workers. A queen can lay up to 1,500 eggs in a single day and may ultimately lay more than 1 million eggs over her 4 to 5 year lifespan. When newly emerged, a worker spends her first 2 days eating pollen and honey. She then spends 3 weeks working inside the hive as a nurse feeding larvae with royal jelly that she secretes from her hypopharyngeal gland. Larvae that are destined to become workers or drones get royal jelly for a few days and then are fed pollen and honey. Larvae that are destined to become queens, on the other hand, are fed royal jelly throughout their development. Nurse bees also produce wax that is used to build new cells and repair the comb. As the worker ages, she switches tasks and becomes a forager. Foragers visit flowers and gather nectar and pollen. Pollen is collected in the corbiculum or pollen basket on the outer face of the hind tibia that is specialized for the transport of pollen and nest-building materials. Nectar is stored in an anterior region of the gut called the honey crop and is regurgitated into storage cells or recipient hivemates upon return to the colony. Such transfer of food among nest members is called trophallaxis. Honeybee workers share information about the location of nectar and pollen-producing plants using a remarkable dance language. Von Frisch 1967. A figure-8 waggle dance of varying speed is performed if the food source is more than 100 meters away. The angle of the waggle run in the middle of the dance relative to the vertical allows the bees to follow a course to the food, relative to the position of the sun. Figure 3.3. If the food is less than 100 meters away, then a round dance is performed. Workers often aggressively defend the hive. They have a barbed sting such that stinging an intruder is basically suicidal. Another method of defense used against intruders such as hornets is for workers to crowd tightly around the intruder and shiver their thoracic muscles, which can generate enough heat to kill the trespasser. When a queen ages, the colony becomes overcrowded or food becomes limited, workers begin to make large queen cells. Just before the first new queen emerges, the old queen and about half the workers leave the colony as a swarm. The first queen to emerge stings unemerged queens and then leaves the hive for a mating flight. The drones compete with each other to mate with the queen and the queen returns to the hive after mating. 3.3.2 Ants. Ants live in colonies ranging from a handful of individuals to hundreds of millions of workers inhabiting underground structures reaching 6 meters below ground. Hodiersis LL. and Wilson 1990. Worker ants are wingless, while queens and males are winged. Queens lose their wings after mating. It is thought that the lack of wings allows workers an advantage when foraging in leaf litter and beneath the soil surface. Chemical communication in ants has reached an extraordinary level. The average ant has more than 40 anatomically distinct exocrine glands involved in pheromone production. Billen and Morgan 1998. Pheromones play important roles in colony regulation, trophallaxis, trail marking, recruitment, recognition of nestmates and defense. Caste is determined by genetic environment interactions. Ant heads are often modified according to caste and can be very large with massive or otherwise specialized jaws. For example, castes of some species can have jaws modified for crushing seeds, blocking nest entrances or dismembering enemies. Figure 3.4. Head shape and mandible structure inform us well on the ecological roles of ants in their environments. The two largest subfamilies of ants are the Myrmicinae and Formicinae. Myrmicine ants have stings and venom while Formicine ants defend themselves by spraying formic acid. Hodiersis LL. and Wilson 2009 contend that leafcutter ants, primarily Acromyrmex and Atta, are Earth's ultimate superorganisms, because they have one of the most complex communication systems, the most elaborate caste system, air-conditioned nests and colonies that contain up to 8 million workers and have a lifespan of 10 to 15 years. To produce such vast colonies, the queens have become the ultimate reproductive machines. A typical Atta queen, for example, can produce 150 to 200 million offspring in her lifetime and store 200 to 320 million sperm cells in her spermatheca. Kerr 1962. The unique aspect of the biology of leafcutter ants is their fungus farming. Leafcutter ants cultivate multiple, distant lineages of fungi, most of them belonging to the family Lepiotaceae, agaricales, Basidiomycota, Muller et al. 1998. The fungal cultivars serve as the primary food source for the ants and are provided with cut leaves. Hodiersis LL. and Wilson 1990. A foundress queen propagates the fungus clonally by carrying inocula in her infrabuccal pocket, a cavity located beneath the opening of the esophagus, during her nuptial flight. After mating, foundress queens establish new colonies by digging chambers in the soil, expelling the fungal pellet that they brought from the natal nest, and initiating the cultivation of their own gardens, which are started by using fecal material provided by the queen. Nest founding occurs within a chamber that remains closed until the first brood is reared, at which point the new workers begin to feed on the fungus and take over the fungus culture activities, including foraging for new leaves. As fresh clips of leaves are brought into the nest, they are cut into smaller and smaller. Station 2 Station 1. Station 3. Dance at. Feeding Station 1 280 N. 180. Pieces and treated with ant secretions before being inserted into the fungal garden. These secretions are part of the elaborate system of enzymes that are produced directly by the ants and by their microbial symbionts. Curry et al. 1999. Fernandez Marin et al. 2009. The enzymes along with weeding behaviors Basson-Charette 1994 keep the garden free of bacterial and fungal pathogens and parasites. The workers transplant mycelia to other parts of the garden substrate and stimulate the fungal garden to grow very quickly. Acromyrmex and Atta have highly polymorphic workers, and this high level of polymorphism correlates with the complex division of labor that exists within their colonies. Wetterer 1994. In some Atta workers, for example, the head width varies 8-fold and the dry weight varies 200-fold. Small workers are required as gardeners of the fungus, large workers are capable of cutting vegetation, and workers of all sizes are capable of brood care. Wilson 1980. Only small workers can take care of the delicate fungal hyphae within the garden, whereas the largest workers with their powerful mandibles designed to cut tough leaves are excellent defenders of the colony. The largest workers are usually recognized as a soldier subcaste or a full caste if really distinct from workers, with a disproportionately large head capsule. It is thought that the queen produces the maximum number of individuals who together can perform all the essential colony tasks. As the colony grows, the range of size dimorphism becomes even more extreme and worker production becomes skewed towards large individuals. Age polyethism is also widespread in leafcutter ants. Young workers tend to perform tasks inside the nest and older workers tend to labor outside the nest. In a few years, the colony can become a physical monstrosity. The central mound of the underground nest can grow to more than 30 meters wide with smaller, radiating mounds extending out to a radius of 80 meters. The whole underground nest can take up 30 to 600 square meters. 3.3.3 Termites. The most widespread, non-hymenopteran eusocial insects are the termites, Higashi et al. 2000. Termites are sometimes called white ants because they're. Figure 3.4 A sample of front views of head capsules of ant species showing the great diversity of shapes and a variety of adaptations for different functions. From Wheeler 1910. The ant below is a Zacryptoceras species which uses its head to block an entrance to the gallery system, as is the case for example L above in a related species. From Ho Diaresis LL Dobler and Wilson 1990. The Colobopsis head capsule in example S above is also adapted for plugging entrances to nests. Workers are pale and their social organization is very similar to ants. Thorne 2003. Termites, however, are not closely related to ants at all, but are instead close relatives of cockroaches. Thorne and Carpenter 1992. All termites live in permanent social colonies that range in size from a few dozen individuals to massive, architecturally complex mounds containing millions of termites. Krishna and Wiesner 1970. Termite colonies typically contain four castes: primary reproductives, the kings and queens, supplementary reproductives, soldiers and workers. Primary reproductives are originally winged, but shed their wings after a short dispersal or nuptial flight. Colonies typically have a single queen and a few reproductive males. Supplementary reproductives become reproductively active if anything happens to the king or queen. Soldiers are sterile males and females with heavily sclerotized and modified heads. Soldiers are often armed with large jaws or the ability to eject sticky secretions from their heads. For example, soldiers of species in the subfamily Nasutidermatinae have frontal glands in their heads that produce sticky and noxious chemicals. The chemicals are sprayed from a pointed nozzle, or rostrum, at the front of the head. The spray is a viscous entangling agent and irritant, capable of quickly hindering the mobility of ants and other predaceous arthropods. Worker termites of the species Globiderm sulphurus take defense to even further extremes. They act as soldiers and have a suicidal self-defense mechanism where they self-destruct by bursting their abdomens and covering their enemies, typically ants, with a sticky secretion. Bordereau et al. 1997. Worker termites can be male or female, resemble nymphs, and usually outnumber soldiers 50:1. Much like social hymenopterans, termite workers are responsible for nest construction and maintenance, foraging and brood care. All termites use symbiotic bacteria, protozoa or fungi to digest cellulose. Many of these symbionts are passed throughout the colony trophallaxis. More detail is provided in Chapter 6. 3.3.4 Thrips, aphids and beetles. 3 other, non-hymenopteran insect orders contain species that have recently been described as eusocial. There are at least 6 species of eusocial thrips, Thyssanoptera, that induce galls on acacia in Australia. Crespi 1992, Crespi et al. 1998. Galls are produced in the spring by single, inseminated, winged Macropterus females. Galling sites on the modified petioles of these plants seem to be in great demand. From gall initiation until gall closure, foundresses use their armed forelegs to fight each other over gall ownership. After a foundress is enclosed in the gall, she feeds on gall tissue and oviposits in the inner gall surface. Newly eclosed offspring feed and develop inside the gall. Adult offspring, both males and females, of the foundress are of 2 discrete types or castes: winged Macropterus individuals and normal, forelegs, and Micropterus flightless individuals with much larger forelegs. The Micropterus individuals act as soldiers and use their enlarged forelegs to attack kleptoparasitic thrips that try to invade the gall, as well as intruders such as ants. The soldiers are effective defenders and can significantly reduce attacks on the foundress. Female soldiers have significantly smaller oocytes than foundresses and broods are often highly femalebious. In the most derived thrips species of this group, the soldiers are almost functionally sterile. Chapman et al. 2002, and the Macropterus females can function as dispersing reproductives and are analogous togines, facultative reproductives, in the eusocial Hymenoptera. Gall-inducing aphids in 2 small subfamilies, the Pemphigenae and Hormiphidae, share a great deal of biology with the eusocial thrips. Aoki 2003, Pike and Foster 2008. These aphids induce galls on their host plants. All members of the gall initially descend from a single foundress, and are therefore clone mates, but later colonies become chimeric, of mixed genetic origin, with intruders from other colonies. In addition to normal first instar nymphs, there are specially adapted first instar morphs called soldiers. Foster 1990. These soldiers cluster around the gall's entrance and actively attack predators, piercing them with their stylets and thickened forelegs. Figure 3.5, often losing their lives defending the gall and protecting their relatives. Foster 1990. During the summer there are about 300 aphids per gall in many species, of which approximately 50% are soldiers. The aphids in the gall feed on phloem and defecate honeydew. In some species, the soldiers actively clean the galls, which increases colony growth. Benton and Foster 1992. A eusocial beetle has recently been discovered. Kent and Simpson 1992. The ambrosia beetle Austroplatypus incompertus lives in galleries in the heartwood of living eucalyptus trees in Australia. Solitary fertilized females initiate gallery systems in the autumn. It takes about 7 months for the female to tunnel the 50 to 80 from the bark through the sapwood into the heartwood. Fungal propagules are distributed from mycangia on the pronotum of the female and symbiotic fungi Ambrosiella sp. Establish in the heartwood and sporulate on the tunnel walls. In the following year, the female lays eggs at one end of the gallery. The larvae, like the adults, eat the fungus and take 2 to 3 years to develop from egg to adult. After 4 years there is a marked change in the social order of established colonies. Around that time galleries start to contain unfertilized adult females that extend and maintain the galleries, remove frass and protect against predators. Females residing in the gallery either lose or remove the last 4 tarsal segments, making it impossible for them to leave the colony. Colonies are extremely long-lived and some are known to persist for up to 37 years. A. S. N. 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 S. 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 B. C. D. Figure 3.5 Examples of soldier aphids, left, and normal siblings, right, belonging to the species Pemphigus spirothecae, Colophina monstrifica and Pseudoregma bambusicola. In Pemphigus spirothecae, A first instar soldier, S, has enlarged legs and a thickened cuticle compared to a normal nymph, N. The soldier of Colophina monstrifica, B, is much larger than a normal nymph, and has relatively longer legs, a shorter rostrum, and more sclerotization. In Pseudoregma bambusicola the soldiers have. Caption for figure 3.5 Cont. Enlarged front legs and heavy sclerotization. The soldier in D also has longer legs and frontal horns used in impaling intruders. The life cycle of the aphid includes 2 host plant species. The primary host soldier, C, is a second instar nymph. The secondary host soldier, D, is a first instar nymph. The scales are 0.5 millimeters. From Stern and Foster 1996. 3.4 Evolution of Sociality, Darwin's Dilemma. Evolutionary biologists have long recognized that altruistic behavior in general, and the presence of sterile workers in particular, is difficult to explain within the concept of individual selection, Sturtevant 1938, Anderson 1984. Darwin was well aware of this problem and noted that sterile workers in eusocial insects posed one special difficulty, which at first appeared to me insuperable and actually fatal to my whole theory. Darwin 1859, p. 236. Darwin noted that if the combined offspring of the queen formed a colony that would allow her to produce more offspring than an otherwise comparable, solitary female, sterile casts would evolve as part of the variation of a single hereditary type. That hereditary type, in this case the colony, not the variable forms it produces, would be the unit of selection, Wilson 2005. Thus, Darwin proposed that workers evolve through selection at the level of the colony instead of through selection on individual fitness. 3.4.1 The haplodiploidy hypothesis. Early evolutionary biologists, however, quickly suggested that selection at the colony level could not fully explain why individuals would give up the possibility of passing their genes to offspring. Anderson 1984, Sober and Wilson 1998. Haldane 1932, 1955 began to formulate a solution to this problem when he observed that altruistic behavior could be favored by selection if altruism increased the success of genetically related individuals, and he proposed that this is likely to be the case in small populations that typically contain many close relatives. Hamilton expanded this idea into a general theory, Hamilton 1964a. The core concept of this theory was expressed in what has come to be called Hamilton's rule, RB greater than C, where B is the benefit to the recipient of the altruistic act, C is the cost to the bearer, and R is the degree of relatedness between them. This simple inequality has been incredibly influential, partly because it is so easy to understand. Hamilton's rule demonstrated that natural selection might work to increase inclusive fitness, the sum of fitness gained through producing offspring, direct fitness, and through affecting the fitness of individuals that share genes with the altruist, indirect fitness, Maynard Smith 1964, Herbers 2009. Hamilton immediately applied this idea to the evolution of eusociality within the Hymenoptera, Hamilton 1964b. Hymenoptera are haplodiploid and females develop from fertilized eggs and males from unfertilized eggs. This can create strange patterns of relatedness within families. For example, a male derives all his genes from his mother and thus R son mother 1/4 1, but the mother gives half her genes to her son and thus R mother son 1/4 0.5. Most importantly, sisters share 3/4 of their genes with each other, R sister 1/4 0.75, but only half with their potential offspring, R mother offspring 1/4 0.5. Thus for haplodiploid species, helping to raise sisters instead of offspring is a good strategy for getting copies of genes into the next generation, figure 3.6. This concept provided an excellent framework for studying the context of cooperation and conflict and seemed like an obvious answer to the eusociality conundrum, Herbers 2009. Hamilton's application of kin selection to eusociality within the Hymenoptera is often called the haplodiploidy hypothesis. Although a few researchers warned against an overemphasis on relatedness and pointed to other factors that could play an important role in the evolution of eusociality, such cautions were basically ignored and this idea quickly dominated the field, Anderson 1984. For example, Wilson 1971 stated that the key to Hymenopteran success is haplodiploidy, and that nothing but kin selection seems to explain the statistical dominance of eusociality by the Hymenoptera. The evidence seemed overwhelming. 10 out of 11 independent origins of eusociality that were known in the 1960s occurred in the Hymenoptera. Some findings, however, slowly began to raise doubts about the overwhelming focus on the importance of haplodiploidy, Anderson 1984. Notably, although all Hymenoptera are haplodiploid, only a very small percentage of Hymenopteran species are eusocial. Moreover, most mites and ticks, many thrips, whiteflies, scale insects, some beetles, most rotifers and some nematodes are haplodiploid, yet eusociality is either completely unknown or incredibly rare within these taxa. 3.4.2 More general views. Termite biology provides a particularly strong challenge to the focus on relatedness, Thorne 1997. Termites originated from cockroach-like ancestors and evolved eusociality about 100 million years ago. All termites are diploid and have sterile casts of both sexes. Hence, the asymmetric degree of relatedness inherent between hymenopteran brothers and sisters, and between their sisters and their offspring, is not generated in termites. A number of hypotheses have been proposed to explain the evolution of termite social biology. For example, termites depend on symbiotic intestinal protozoa for digestion of cellulose, and Cleveland et al. 1934 suggested this symbiotic relationship predisposes termites to social life. The protozoa are lost at each molt, so developing termites must obtain new protozoa through transfer from other colony members. Hence, the symbiosis with protozoa necessitates social life until adulthood. It has been noted, however, that passing of symbionts does not require advanced sociality. Extended parent-offspring contact generated by a simple social system allows for transfer of symbionts in cryptocercidwood roaches, the closest extant relative soft termites. Thorne 1997. Hamilton 1972 proposed that inbreeding could lead to high degrees of relatedness within colonies and facilitate the evolution of eusociality. The problem with this idea is that inbreeding will, over time, also increase the relatedness of parents to offspring and not just among siblings, thus providing little or no selection fourth worker caste to forego reproduction. Thorne 1997 carefully examined the many termite hypotheses and concluded that it is unlikely that eusociality in termites arose as a result of evolutionary forces acting on any one dynamic or on any single life history component. Instead, she proposes that a suite of ecological and life history traits of termites and their ancestors predisposed. Figure 3.6 The degrees of relatedness within a colony of haplodiploid insects such as ants, bees and wasps, if the king and queen are monogamous. Sisters, on average, share 75% of their genes because each sister gets identical genes from their haploid father. This means that sisters can produce more copies of their genes if they help rear sisters instead of their own offspring. In diploid species, sisters, on average, share 50% of their genes because they have a 50% chance of getting the same genes from their father and mother. Based on Trivers and Hare 1976 and Hamilton 1964b. Them for eusocial evolution. These characteristics include familial associations in cloistered food-abundant habitats, slow development, overlap of generations, monogamy, iteroparity, high-risk dispersal for individuals, opportunities for nest inheritance by offspring remaining in their natal nest and advantages of group defense. Table 3.2. In this table Thorne divides factors into ecological, behavioral and life history categories: nesting habitat, parental care, development, genetics, mating system, reproductive cycle, dispersal and founding nests, inheritance and defense. Each of these factors are explained briefly in the table. However, in real life several factors would combine to promote the evolution of eusociality. Thorne 1997. For instance, primitive termites live under bark where food is. Table 3.2 Some ecological and life history characteristics of eusociality. Modified from Thorne 1997. Nesting habitat. Claustral family associations, e.g., enclosed in a cavity. Safe, initially small, and persistent habitats, with opportunities for expansion and multi-generational occupation. Nesting in enclosed spaces keeps relatives close and fosters kin selection, and foraging by immatures for each other and the reproductives. Parental care, subsociality. Family groups development time, life cycle. Slow development and long generation times with overlap of generations. Long lifespan, particularly in reproductives gradual metamorphosis so helping in the next generation can begin as immatures soon after egg hatch, in termites. Genetics. Haplodiploidy. Females are more related to sisters than their progeny, so helping the queen to raise sisters increases fitness, in Hymenoptera. Mating system. Single father, monogamy in haplodiploid groups female progeny share 3/4 of their genes, with likely selection for helping a mother to raise other female progeny, see figure 3.6. In diploid species monogamy results in half of an individual's genes being shared with siblings and with progeny, so helping to raise siblings is as genetically profitable as producing progeny. Reproductive cycle. Iteroparity. Repeated bouts of reproduction enable offspring to care for new siblings. High risk and/or time investment in dispersal and founding of new nests. Remaining in the parental nest as a helper is likely to be safer than dispersal and establishment of a new nest. Opportunities for replacing reproductives. A mature offspring may take over nest as the reproductive caste, or act as a supplementary reproductive. In either case the individual inherits nest resources. Defense. Group defense of rich resources in the nest essential. Group defense against competitors. Important specialized defense is effective, employing workers and soldiers, use of stings, venoms, biting, etc. Plentiful but of low nutritional quality. The nest site is well protected, so a founding queen can remain at this site for a long time while laying eggs, and family groups interact intimately. With hemimetabolous development, the nymphs can act as workers to aid the queen. The life cycle is prolonged because of low food value, and possibly because of the protected nest site, resulting in a likely overlap of generations and cooperation among generations. Monogamy results in a 50% genetic relationship among siblings and a 50% relationship with progeny, so breeding itself or helping siblings are genetically and fitness neutral, but with a significant advantage to staying in the nest and avoiding the dangers of the outside world. 3.4.3 Sequential trait accumulation. This multifaceted array of factors provides a logical start to figuring out how so many traits could be selected for, and in what order they were assembled into the life history and behavior of a social insect species. An intriguing approach was adopted by Hunt 1999, 2007, who argued that tracing essential characteristics of a social group back to their phylogenetic origins will recapture the stepwise aggregation of salient traits. Tracking the evolution of the social wasps, Hymenoptera. Vespidi, the accumulation of traits necessarily started with the most primitive groups in the order, figure 3.7. Hunt mapped salient traits on the phylogeny of the Hymenoptera. An abbreviated treatment is provided here. Trait 1 included at the base of the symphyta, sawflies, woodwasps, etc. The possession of mandibles, a lepismatid type ovipositor, first seen in the primitive silverfish, Thysanura, lepismatidae, membranous wings, and the presence of haplodiploidy. Mandibles became important in social wasps in nest building and provisioning the nest. The ovipositor evolved into the sting of wasps, critical in the defense of the colony. Membranous wings enabled strong flight for foraging and the carrying of heavy insects as food for larvae. Haplodiploidy resulted in closely related sisters as workers. At branch 2, carnivorous larvae evolved from their herbivorous ancestors. At branch 3, larvae become legless in the parasitoid wasps and the larval hindgut becomes closed, meaning ultimately that they do not foul the nest. A basal trait of the Apocrita is the petiolate abdomen, in which the thread waist of the adult limits nutrition to liquids, although masticated insects can be fed to larvae in the nest. At branch 4, the oviduct becomes separate from the ovipositor, such that the ovipositor can evolve into a sting and associated glands become modified to produce venom and pheromones. The sting and venom enable anesthetization of prey and their storage in a nest. At the origin of the Vespidi, at branch 5, nest construction and rearing of larvae in the nest evolve, with provisioning by the queen and workers. Additional steps are described by Hunt to conclude the list of the most important traits in the social wasps. This approach shows how traits originating with the primitive herbivorous insects, parasitoids and predatory solitary wasps all contribute to the evolution of eusociality of the Vespidi. In this lineage, behavioral, genetic and ecological traits blend into the evolutionary scenario. They show how many aspects of biology need recognition as contributing to the emergence of a social insect group. 3.4.4 The continuing debate. A growing number of biologists are beginning to rethink the focus on haplodiploidy and relatedness in the evolution of eusociality, and this has ignited a vigorous, ongoing debate. Wilson and Ho Diaresis LL 2005a, Foster et al. 2006, West and Gardner 2010. Their evaluation of the importance of haplodiploidy has largely been driven by new discoveries of eusocial animals. Many additional eusocial insects have been discovered, including eusocial aphids, thrips and beetles, see section 3.3.4. Other invertebrate eusocial species have also been identified, including eusocial sponge-dwelling shrimp, Duffy 1996, and a eusocial tanglefoot spider, Valrath 1986, Ripstra 1993. Perhaps most surprising, 2 species of mole rats were found to be eusocial, the first discovery of eusocial mammals, Jarvis 1981, Jarvis and Bennett 1993. In several cases, these discoveries involve multiple species within the group and multiple independent evolutionary origins of eusociality, e.g., Jarvis and Bennett 1993, Duffy and McDonald 2010. Because most of these groups are diploid, the association between haplodiploidy and eusociality is no longer statistically viable, Wilson 2005, Herbers 2009. This does not mean, however, that high levels of relatedness are unimportant in the evolution of eusociality. Indeed, many authors still believe that close relatedness is crucial to the evolution of eusociality. For example, there has always been some dispute in the literature of whether close relatedness is a prerequisite for the evolution of eusociality or a consequence of eusociality. Hughes et al. 2008 tested the hypothesis that the evolution of monogamy, in this case breeding with a single male, menandry, evolved within eusocial bees, wasps and ants prior to the evolution of eusociality. The idea is that monogamy increases relatedness among offspring, thus increasing the potential for kin selection to act. They found that monogamy was the ancestral state for all 8 independent origins of eusociality that they examined. Hughes and colleagues argued that their findings strongly support the hypothesis that high relatedness was important in the evolution of eusociality because monogamy and the evolution of eusociality are strongly linked. Likewise, Duffy and McDonald 2010 found that eusociality only arose in sponge-dwelling shrimp species with non-dispersing larvae that form tight family groups, suggesting that close relatedness was strongly correlated with eusociality in this group. There have also been renewed calls for a focus on the role of relevant costs and benefits in the evolution of eusociality, rather than relatedness. Thorne 1997, Herbers 2009. Anderson, 1984, was an early critic of the focus on relatedness and suggested that studies evaluating the costs and benefits of brood care and defensive nests or protected cavities and the low success of young adults or solitary pairs that attempt to reproduce on their own would yield important results. Likewise, Queller and Strassman, 1998, have proposed that fortress defense, the fortress being a domicile that provides protection and food, and life insurance, brood care by non-parents when parents are killed or otherwise unable to rear offspring, are key benefits of eusociality. Life insurance will be particularly important for species where the brood have to be continuously provisioned during development, Queller 1994. These analyses, however, are still conducted within the framework of Hamilton's rule, RB greater than C. The idea is that if the benefits are relatively high and the costs are relatively low, then eusociality will be favored even if relatedness is not very high. There are substantial data to support this viewpoint. For example, the probability of foundresses successfully starting a new colony is incredibly low in ants, bees and termites, Ho Diaresis LL and Wilson 1990, 2009, Thorne 1997. Studies of facultatively eusocial bees have been particularly insightful. R. Smith et al. 2007 found that solitary nests of the sweat bee, Megalopta genilus, suffer significantly higher levels of failure than eusocial nests and produce significantly fewer broods. There has also been a renewed focus on group selection as an explanation for the evolution of eusociality. Wilson, once a strong advocate for the importance of kin selection, has proposed a multi-level selection model that promotes group selection as the primary binding force that favors eusociality. Wilson 2005, Wilson and Ho Diaresis LL 2005a, Wilson and Wilson 2007. Group selection, the differential survival and reproduction of entire cooperative groups, was once a widespread explanation of adaptations, but its importance as a force of evolution has been controversial for over 40 years, e.g., Williams 1966. In Wilson's model, close relatedness is a consequence of eusociality, not a causative factor in the evolution of eusociality. Wilson's proposal has been strongly criticized as misinterpreting kin selection theory, Foster et al. 2006, and for largely being a semantic argument, Shavit and Milstein 2008. It will be interesting to see if Wilson's latest ideas can be developed into a set of stable hypotheses that will alter widely held views of the evolution of eusociality. 3.5 The ecological consequences of sociality. Social insects are often dominant species in the ecosystems in which they live. Ants, for example, have been one of the most diverse, abundant and ecologically dominant animal groups for over 50 million years. Ho Diaresis LL and Wilson 1990, Wilson and Ho Diaresis LL 2005b. They comprise 10 to 33% of the biomass of many ecosystems and can reach densities of 8 million per hectare in tropical forests. Wilson 1990. It is estimated that their biomass in the Brazilian Amazon is 4 times greater than all of the terrestrial vertebrates combined. Fitkow and Klinge 1973. Their ecological success is a function of their sociality. Individual ants within the colony can specialize in particular tasks, and groups of workers can quickly switch among tasks such as colony maintenance, to group foraging, to colony defense, etc., a major advantage over solitary insects. Ants are classified into a single family, the Formicidae, and comprise 16 subfamilies, 296 genera and over 10,000 species. Bolton 1994. They have a huge geographical range and live in an incredible array of habitats. Ants are among the leading predators of other insects and small invertebrates and are major herbivores and seed predators in many habitats. Omnivorous ants in Polish meadows, for example, can consume up to 3% of the primary production and 40% of the prey biomass available each season. Folger 8 1998. An average size colony of Formica polyctena can consume 106 insects and 155 liters of honeydew from just a quarter hectare area during one growing season. Horstman 1974. Leafcutter ants can remove 17% of the annual leaf production of a tropical forest. Charette 1989, and one at a nest can consume 1 to 2 tons of fresh leaf material per year. Folger 8 1998. Ants are also major ecosystem engineers. Folger 8 1998, McMahon et al. 2000. For example, they can move tremendous quantities of subterranean soil to the surface. The activities of desert ants may lead to soil turnover rates as high as 420 kilograms per hectare per year in Australia, Breeze 1982, and 842 kilograms per hectare per year in the USA, Whitford et al. 1986. Camponotus punctulatus ants move up to 2,100 kilograms of soil per hectare while constructing their mounds in Argentine pastures. Folger 8 1998. Indeed, ants are considered second only to earthworms as biotic sources of soil turbation. Budents have a broader geographical range on Dare, thus more important in many areas of the world. Patton et al. 1995, Folger 8 1998. In the tropics, for example, leafcutter ants are the most important agents of soil modification. Alvarado et al. 1981, and just own colony of Atta sextans in Brazil deposited over 40 tons of soil in 1 year. Folger 8 1998. Ant activity can also strongly affect nutrient and energy fluxes within soil. Nests of Atta columbica in Panama increase fluxes of 13 chemical elements 38-fold in comparison to surrounding areas of forest, probably due to the greater root activity close to the ant nests. Haynes 1978. In Puerto Rico, leafcutter ants have been shown to increase net plant productivity by 1.80 kilocalories per meter too, probably because ant activity increases the availability of phosphorus in the soil. Lugo et al. 1973. Ants can also have dramatic effects on the composition of plant communities via soil and seed dispersal. Horvitz and Shemsk 1986, Wilson 1992, McMahon et al. 2000. A 10-year experiment in the Chihuahuan Desert of southeastern Arizona found that tharbester ant removal resulted in a plant community dominated by small seeded plant species. Sampson et al. 1992. This change in the plant community reflected the harvester ant's preference for large seeds. As noted in Chapter 6 and in Section 3.3.2, many ants form facultative or obligate mutualisms with an incredible range of organisms including fungi, plants and other insects. In many systems, these mutualisms can strongly influence the ecological effects of ants. Many ants, for example, form food for protection mutualisms with aphids, scales and other, honeydew-producing insects. Way 1963, Buckley 1987. In these mutualisms, ants provide protection against arthropod predators and other natural enemies in exchange for carbohydrate-rich excretions from the herbivorous insects. Not surprisingly, these mutualisms often result in dramatic reductions in the number of predators on plants that host honeydew-producing herbivores. Kaplan and Eubanks 2005, for example, found that cotton aphid fire ant mutualisms dramatically increased the number of foraging fire ant workers on cotton plants and that fire ant workers killed over 75% of the predators on aphid-infested plants. It turns out that ant workers that tend honeydew-producing insects often kill herbivorous insects as well as predators. Messina, 1981, for example, found that Formica ants that tend sap-feeding treehoppers on goldenrod plants, Solidago, not only protect the treehoppers from predators, but also kill leaf beetles that are major defoliators of the plant. Likewise, fire ants that tend cotton aphids also kill caterpillars and true bugs that feed on cotton plants. Kaplan and Eubanks 2005. These ant hemipteran mutualisms ultimately result in broad changes in the arthropod community associated with the hemipterans host plant, Wimp et al. 2005. As such, these mutualisms can be considered keystone interactions that change the abundance and distribution of many other arthropods, the effects of which cascade down the food web to affect primary production. Eubanks and Stiersky 2006, Chapter 13. In many cases, these changes can actually increase plant fitness. In both examples detailed above, goldenrod and cotton, the defoliators that were killed by hemipteran tending ants, leaf beetles and caterpillars, were more important herbivores of the plants than the treehopper sand aphids. The mutualisms, therefore, led to increased suppression of these more damaging herbivores and corresponding increases in plant fitness. Messina 1981, Stiersky and Eubanks 2010. A recent review has shown that many ant hemipteran mutualisms indirectly increase plant fitness in this manner. Stiersky and Eubanks 2007. Mutualisms involving ants can even shape the ecology of the world's largest terrestrial herbivores. For example, Madden and Young 1992 studied the ecological consequences of the mutualism between the swollen thorn Acacia drepanolobium on the plains of Kenya and an ant in the genus Crematogaster that nests on these trees. The trees provide the ants with a protected chamber, the swollen thorn, for nesting and nutrient-rich extrafloral nectar for food. See Chapter 6 for a detailed description of the ecology of myrmecophiles. In turn, the ants provide protection against herbivora. Madden and Young found that Crematogaster ants were highly effective at defending drepanolobium trees from foraging giraffe calves. They found a very strong, negative relationship between giraffe browsing of acacia trees and the density of swarming ants on the trees. Although termites have far stricter feeding habits than ants and occupy fewer niches in ecosystems. A. B. 35.0 30.0 25.0 20.0 15.0 10.0 5.0 0.0 Mound. Termites have profound ecological effects. They consume up to 1/3 of the annual production of dead wood, leaves and grass in many habitats, and their densities can surpass 8,000 per square meter. Marcius 1994, Pierce 1997. Termites can act as ecosystem engineers that influence the physical characteristic of soils and associated habitats and strongly influence biogeochemical cycling. Jones 1990, DeCans et al. 2001. A fascinating example of the ecological role of termites occurs in the Kenyan savanna inhabited by drepanolobium trees. Using observational data, large-scale experimental manipulations and analysis of foliar N. Brody et al. 2010 found that drepanolobium trees growing at the edge of termite mounds were more likely to reproduce than those growing farther away, in off-mound soils. Figure 3.8. Although vertebrate herbivores preferentially use termite mounds, long-term exclusion of mammalian grazers did not significantly reduce drepanolobium fruit production. Leaf-in was significantly greater in trees growing next to mounds than in those growing farther away. Thus, soil enrichment by termites was of primary importance to fruit production near mounds. Pringle et al. 2010 found that these termite mounds were not only hotspots of plant growth, primary production, but also local hotspots of animal abundance. Insect abundance and biomass decreased with distance from the nearest termite mound, as did the abundance, biomass and reproductive output of insect-eating predators. Figure 3.9. They also found that the evenly spaced distribution of termite mounds produced dramatically greater abundance, biomass and reproductive output of consumers across all trophic levels than would be obtained in landscapes with randomly distributed mounds. Thus, termites are critically important ecosystem engineers in this system whose distribution plays a fundamental role in ecosystem function. 4. 3. 2. Applications. Social insects as saviors and pests. Honey produced by honeybees was the only sweetener available to humans for centuries, and honey is still a widely used product with an annual value of $1.25 billion United States dollars. Van Engelsdorp and Meixner 2010. As detailed in Chapter 6, however, bees and other social insects are incredibly important pollinators of crops and wild plants. By far the most important contribution honeybees make to modern society is the pollination services that they provide. Of the 115 leading global food commodities, 52 depend on honeybee pollination for either fruit or seed set. Klein et al. 2007. 5 additional commodities would have 90% yield reductions without honeybees, and yield of 16 other commodities would be reduced by 40 to 90% without honeybees. Smaller reductions would occur in at least 19 additional commodities. In total, 14-23% of all agricultural production is directly reliant on insect pollination, and 35% of the human diet is thought to benefit from insect pollination, with honeybees being particularly important. Klein et al. 2007, Van Engelsdorp and Meixner 2010. The global value of insect pollination was recently estimated at $212 billion United States dollars. That represents almost 10% of the total value of agricultural production. Van Engelsdorp and Meixner 2010. Honeybees and the pollination services they provide have been of great recent interest due to colony collapse disorder. Colony collapse disorder is characterized by the rapid loss of adult worker bees from affected colonies, as evidenced by weaker dead colonies with excess brood relative to adult bees, noticeable lack of dead workers, and food stores that are not robbed by hive pests or kleptoparasitic bees. Van Engelsdorp et al. 2009. In the fall of 2006, U.S. beekeepers reported losses of 30-90% with symptoms consistent with colony collapse disorder. This was followed by 2 more winters with significant colony losses associated with colony collapse disorder. Van Engelsdorp et al. 2007, 2008. Many hypotheses have been proposed and at least partially tested to explain the colony losses, including honeybee parasites e.g., varroa mites and honeybee tracheomites pathogens e.g., bee viruses and Nosema microsporidia pesticide residues especially neonicotinoid insecticides, and inbreeding, Van Engelsdorp et al. 2009, Radnieks and Karek 2010. The latest research on colony collapse disorder suggests that an interaction of factors likely explains colony death, although pathogens, particularly viruses and Nosema, appear to be critically important. Cox Foster et al. 2007, Johnson et al. 2009, Radnieks and Karek 2010. Many social insects are also economically important predators of crop pests. Way and Koo 1992. In fact, the manipulation of the weaver ant Oecophila smaragdina to control citrus pests is the oldest known example of biological control and was first documented in 304 AD, Huang and Yang 1987. Weaver ants get their name from their habit of binding leaves and twigs together with silk to form tight nests. The ants spend the night in the nests, but forage outside the nest for their insect prey during the day. These nests have been sold to farmers in Chinese markets for almost 2,000 years, Huang and Yang 1987. To take advantage of these ants, a farmer secures a nest on one tree and then connects it to adjacent trees with bamboo strips. These bridges enable the weaver ants to travel among and build nests on neighboring trees. Eventually the whole orchard can be colonized by O. smaragdina. These ants were critically important biological control agents of citrus and other fruit pests in ancient China and are still extremely valuable in pest management today. For example, weaver ants currently provide economically important biological control in citrus in China and Vietnam, cashew and mango in Australia, and coconut and cocoa in the Solomon Islands and Africa, Van Mealy 2008. Many other ants are also important predators of crop pests. For example, twig nesting ants are highly effective at reducing the abundance of leaf miners, De La Mora et al. 2008 and coffee berry borers, Larson and Philpott 2010, in coffee. Recent studies have also documented the importance of ants as important predators of pests in cotton, Stiersky and Eubanks 2010, Apple, Matthews et al. 2004, Collards, Harvey and Eubanks 2004, Pecan, Ellington et al. 2003, and Rice, Way et al. 2002, and this is just a small list of the crops where ants play a fundamental role in pest suppression. Social insects, however, are definitely double-edged swords when it comes to impacts on human society. Invasive ants, even some of the same ant species that are important predators of pests, can be brutally important invasive species. Many ant species, for example, are invasive species that have devastating ecological effects, Hallway et al. 2002. The red imported fire ant, Solenopsis invicta, is a well-known example of an invasive ant in North America, Vinson 1997, Chinkle 2006. Red imported fire ants were accidentally introduced from Argentinito, Alabama approximately 75 years ago, probably in soil ballast discharged from cargo ships arriving from Brazil, Vinson 1997. They have since spread rapidly throughout the Gulf Coast states, east to the coastal areas of South Carolina, North Carolina and Virginia, and west to California, where they continue to expand their range northward, Vinson 1997, Korzukin et al. 2001. Red imported fire ants have also recently been introduced in Taiwan, China and Australia, Morrison et al. 2004, Chen et al. 2006, Zhang et al. 2007. Because they are broadly omnivorous, extremely aggressive and often superabundant as a result of tremendous reproductive potential, red imported fire ants negatively impact invertebrate and vertebrate communities in both natural and managed ecosystems, Vinson 1994, 1997, Wojcik et al. 2001, Hallway et al. 2002. It is thought that escape from co-evolved competitors, pathogens and parasites, especially parasitic phorid flies, is responsible for their large populations and rapid range expansion in North America, Porter et al. 1992, 1997, Orr et al. 1995. It is also believed that the evolution of polygyny, multiple queens per colony, after introduction and its associated reduction in intraspecific territoriality contribute to the high densities and rapid spread of red imported fire ants, Ross and Keller 1995, Ross et al. 1996. The evolution of reduced intraspecific competition appears to be an important mechanism contributing to several ant invasions. Argentine ants, Leinpithema humile, for example, have evolved reduced intraspecific aggression in North America and are far less territorial than Argentine ants in their native range, Hallway et al. 2002. The corresponding decrease in intraspecific competition leads to faster colony growth, higher Argentine ant densities and faster spread of Argentine ants. Termites are notorious pests of buildings, Sue and Sheffran 2000. Approximately 200 of the 2,300 termite species in the world are known to damage buildings and 83 species cause significant economic losses. The global economic impact of termites is unknown, but annual control costs in the United States are over $1.5 billion. Insecticide barriers are typically used to control soil-borne subterranean termites and slow-acting baits for subterranean termites in and near structures. Whole structure treatments such as fumigation or heat and local treatments such as insecticide injection are the primary control methods for treewood termites. The Formosan subterranean termite, Coptoderms formosanus, is a particularly damaging termite pest, Laxon Osbrink 2003. This termite was most likely introduced into the USA when thousands of tons of wooden military cargo such as crates, pallets and dunnage were shipped from the Asian theater to the USA following World War II. New Orleans, Louisiana, was one of the most active ports of entry for these shipments and now hosts one of the densest populations of Formosan termites in the world. This termite lives in large colonies that may contain over 1 million workers that can cause rapid and significant damage to wooden structures, living plants such as oak trees and even non-cellulosic materials such as insulation on buried electrical and telephone cables. The Formosan termite causes over $300 million United States dollars annually in damage and control costs in New Orleans, and these voracious termites are spreading within the USA, Woodson et al. 2001. Summary. Social insects are fascinating animals that vary in complexity from small groups comprised of individuals of the same age, to species with intricate parental care, to species of superorganisms that have queens, kings and polymorphic non-reproducing workers. Social behavior is widely distributed among insect taxa, although the evolution of a non-reproducing worker caste has been limited to a few orders: Isoptera, Thysanoptera, Hemiptera, Coleoptera, Hymenoptera. The evolution of non-reproductive workers was a long-standing conundrum in evolutionary biology. Hamilton's kin selection theory provided a simple and elegant explanation of the evolution of non-reproducing castes. Despite recent challenges, kin selection, especially when the costs and benefits of eusociality are fully considered, is still the best explanation of the evolution of eusociality. Social insects are often dominant species in the ecosystems in which they live due to their high population density and polymorphic workers. They are major predators of arthropods, very important seed predators and dispersers, and ecosystem engineers that play important roles in soil dynamics and pollination. Social insects form facultative or obligate mutualisms with an incredible range of organisms including fungi, plants and other insects, and these mutualisms are often keystone interactions that dictate the abundance and distribution of other species within the community. Social insects also have strong effects on humans. At least 10% of the total value of global agricultural production relies on social insect pollination of crops, and social insects like ants are vitally important predators of insect pests. Social insects are also important pests. Invasive ants, wasps and termites, for example, are often urban pests of buildings and landscapes and have had terrible impacts on wildlife such as ground nesting birds, reptiles and amphibians. Termites are often devastating pests of houses and other structures. In total, social insects cause billions of dollars in damage and control costs every year and are typically considered some of the most damaging pests in the world. Questions and discussion topics. 1. Wilson. Part 3. Species interactions. Contents. Chapter 4. Plant and herbivore interactions. Chapter 5. Lateral interactions, competition, amensalism and facilitation. Chapter 6. Mutualisms. Chapter 7. Prey and predator interactions. Chapter 8. Parasite and host interactions. We build from Part 2 on behavioral ecology, which is devoted to interactions among individuals and within social groups, to interactions between species and trophic levels. Gradually we move up the trophic system, starting with plant and herbivore associations, and then to interactions among herbivores involving competition, strong asymmetric interactions and facilitative interactions. After this, in the next 3 chapters, 6 to 8, we return to interactions between trophic levels, treating mutualistic relationships, the interplay of prey and predator, and of host with parasite. In aggregate, these kinds of relationships constitute the main forms of interactions to be found on any landscape. Plants are the major primary producers in any ecosystem. They set the stage for interactions passing up the trophic system in a community. Therefore, the insect ecologist needs to understand plants as food and habitat for insects, and their bottom-up role in plant and insect assemblages. This point of view justifies considerable detail on plant and herbivore interactions in a relatively long chapter on this topic. Understandably, the subject is of great interest to ecologists with a resultant large literature and many hypotheses, for the subject is fundamental for understanding insects in nature, in agriculture, forestry, horticulture, biological control and conservation. As we saw in Chapter 2, plant characteristics can impact trophic levels above the herbivores because toxins, attractants and repellents may impact predators, parasites and mutualists. These interactions considered here are the building blocks of communities and ecosystems. Their understanding is necessary as we progress from this part to the subsequent parts of the book, which deal with larger scale and more complex arrays of interacting species. Part 4 on population ecology, Part 5 on communities and Part 6 on broad patterns in nature. For example, the population dynamics of a species may be profoundly influenced by food supply, competition and natural enemies, and the other interactions discussed here in Part 3 all have a role to play in community structure. Therefore, the contents of this part develop in a logical way the understanding of insect ecology. 4. Plant and herbivore interactions. In their classic paper entitled, Butterflies and Plants, A Study in Coevolution, Ehrlich and Raven, 1964, envisioned an arms race between plants and herbivores, whereby each player exerted reciprocal selective pressure on the other that resulted in evolutionary change. Thus, the arms race between plants and herbivores emphasizes an ongoing reciprocal interplay with plants erecting defenses, herbivores breaking defenses with novel offenses, plants countering with new defenses and so on through evolutionary time. Mitter et al. 1991, Herrera and Pelmer 2002, Thompson 2005. Moreover, breakthroughs in plant defense or herbivore offense are thought to create adaptive zones that promote speciation, lineage diversification and thus the generation of biodiversity. However, an apparent dilemma arises because plants in general have longer generation times and lower recombination rates than their insect herbivores, and especially plant pathogens, which should hinder their ability to keep pacing the evolutionary arms race. Witham 1983. Yet, plants have clearly done so. While the concepts of arms races and coevolution are useful as an overall theme in this chapter, we do not wish to imply that coevolution is a general phenomenon in the interaction between plants and insect herbivores. Strong cases can be developed for coevolution among mutualists, Chapter 6, and parasite-host relationships, Chapter 8, but there is a shortage of sound evidence that insect herbivores have impacted plant traits in a coevolutionary manner, Futuma and Agrawal 2009. In this chapter, we will explore the issue of plant-herbivore interactions in much greater depth and attempt to resolve the apparent disadvantage of plants in their arms race with herbivores. Before doing so, however, we need to learn far more about the players themselves, examine the effects of plants on herbivores at the individual and population levels, and discover more about the complex world of plant-insect interactions. Toward this end, we will elaborate on the incredible taxonomic and ecological diversity of insect herbivores and their variable feeding styles, investigate the barriers e.g., nutrition and allochemistry that plants pose to herbivore attack, explore the counterploys herbivores have evolved to overcome plant defenses, visit plant defense theory, determine how herbivores and plants affect each other's distribution and abundance, and examine how such information might be